me now.
He gets.
But I will totally go.
All right, welcome back, everyone. Well, I'm happy to say that our audio has gotten better, so there won't be any more people trying to get their audio to work. So Rick's got a Bluetooth huge speaker, so we're definitely now better on the audio front. Well, welcome back. We're going to start now with something that used to be, or still is, very close to my heart, to corals. And we're going to go with Rick Williams. Tell us about restoring coral reefs at scale.
All right, thanks everyone, and thanks David for inviting me. Seems to me a little bit off topic from most of what you guys are doing, but I think we've done some pretty interesting work at Coral Gardeners applying AI, developing some technologies which they're using in the field. And so, well, I hope you find it all interesting. I'm going to start out by talking a little about who Coral Gardeners are, what we're doing with tech there, and a little bit about my background, and then I'll dig into the details. And come on, slide, here we go. So Coral Gardeners is it's a coral reef restoration organization based in Moorea, and the island next to Tahiti in French Polynesia. It's 8 years old. It was founded by Titouan Bernico, and who was born and raised in French Polynesia. And Titouan is now 27 years old. So you can see he was pretty young when he decided that his life mission was going to be to work on coral reef restoration. Basically, he saw the reefs that he surfed on and dove on degrading, and wanted to figure out what he could do about it. So Coral Gardeners' mission is basically coral reef restoration, very much at a community level, small scale, and with a vision of creating jobs for local people. Definitely not a large-scale project. We're very, I'd say, complementary to the larger scale efforts which are going on in places like Australia and Saudi Arabia. But with that said, in 8 years, the organization has grown to have the branch based in Moorea, a couple of other sites in French Polynesia, a branch in Fiji, and a branch in Thailand, and about 75 full-time employees. So it's definitely grown a lot. What I'm going to be talking about today is mostly work that was done on Moorea, working out of the headquarters. So Moorea is about a 30-minute ferry ride from Tahiti. It's a stunningly beautiful place to get to go to work. In that image that you see on the right-hand side over here, there's a floating platform with solar panels on it. And you'll see what that gets used for a little later on, as we have some power technology in the water there monitoring the nurseries. And you can see these, you can lightly see these rectangular structures in the water here and here. And these are all coral nurseries. I'll show you in a little more detail in just a minute, so that you understand a little about what the process is. I think understanding the process and understanding the system is a super important part of actually getting AI tools to work to work well for us. This is Coral Gardeners' mission statement. It's to revolutionize ocean conservation and create a global movement to save the reef. As you can see, Titouan is an ambitious man. In this image, this is looking into Cook's Bay. The Coral Gardeners headquarters is just over here, and a lot of the work that I will be showing you was done on a restoration site just out of the image over on the left-hand side there at a place called Tiaya. So what does it mean to revolutionize, and what's the vision of how to revolutionize? I think there's basically three things going on. One is, as I was talking about, the locally based restoration work, and thinking of coral reef restoration as a profession, as work for people who perhaps used to be fishing on the reef, and that livelihood is no longer available because of the degradation of these ecosystems. Along with that is supporting that work with cutting-edge technology to make the work as efficient and productive as possible. That means developing technologies which are deployable and appropriate to the people who are working out in the field and working in close collaboration with the people in the field. But the third piece of it, and some of you might have heard of Coral Gardeners before, is the outreach. Coral Gardeners really has global outreach. They have over 1 million Instagram followers, so they have, I think it's over 1 million now, it's very close to 1 million. And so they basically have an internal media house that develops content that is just stunningly beautiful, and I'm just going to plug it. You really should go look at some of their videos. It's really quite amazing what they do. So I want to talk a little about what coral gardening is, so that what I talk about later on will make sense to you guys. The idea is to grow corals and replant them onto degraded areas of the reef. First step of that is figuring out where to replant. You need to find you need to find areas where there has been reef degradation, but they're not under so much environmental stress that, if you plant stuff out, it'll just die again. And so there's definitely a lot of monitoring work that goes into that. The process starts with finding healthy coral colonies which are fairly local to the site that you want to restore, harvesting small fragments of coral from those, placing them. It's very simple, taking a piece of rope and untwisting, putting the coral in and retwisting, and building these ropes of corals from a single colony, and then mounting them on these nurseries you see in the picture here. There's just a rebar. This is only part of a nursery. There'll be a second piece of rebar down here and probably a third one along, and then tying the ropes between the pieces of rebar, which are in the lagoon in Moorea. This method is used in Moorea because the lagoon is it's a large, shallow water environment with a sandy bottom. It's fairly easy. Well, easy. I would never do it, to go in the water with your scuba gear on and sledgehammer the rebar into the lagoon floor, etc. Finally, after these coral fragments grow, these large pieces are probably 3 years old. Some of these smaller ones are probably 1 year old. Once they get to a large enough size, they're outplanted back out onto the reef. And throughout this process, from the harvesting from the donor colony, from building the ropes, and then planting back out onto the reef, the fragments are tracked, measured, and then after the reef has been had the restoration work done on it, the reef is measured repeatedly in order to track the progress of the outplanting. And so all of that in-water work, a lot of in-water data collection, and that is really the target of a lot of the technology that we're working on developing. There's a group within Coral Gardeners, call ourselves CG Labs. It was started by Drew Gray, who came from the self-driving car world, but he is just an amazing jack of all trades, loves computer vision. We do a lot of computer vision work. On the left is Hannah Stewart. She's our head of science. She's a longtime coral reef ecologist. Me on the right, and then down on the bottom, Mark is an iOS developer. Peter is a sort of machine learning software pipeline person. He also works at Conservation X Labs. He works for us 30% time. And Sophia is I think she's about to start a PhD program in Hawaii. Her specialty is 3D modeling on the reef, so photogrammetry and data extraction from the 3D models that photogrammetry produces. And me, I'm kind of a jack of all trades. I call myself a computational ecologist. That's kind of where I sit. I've done a lot of computer software work, some pretty intense commercial software development projects. I have a PhD in physical oceanography, but pretty much all of my academic work has been in ecology. I call myself a theoretical ecologist. So not a lot of field work in my background. This is by far the most applied and field-oriented thing I've ever worked on. And yeah, it's been great fun so far. So some of the technologies that I'll be talking about today, two main technologies, a tool that we call Reef app. So it is an iPhone app which is designed for doing all of the in-water data entry and data collection. Before this, Coral Gardens was working like an awful lot of marine biologist work. You go out with your scuba gear on, you have what they call a slate, which is a sheet of plastic and a waterproof pencil, like a grease pencil, and you write on the slate. And then you come on shore, and after your day of being in the water diving, you have to sit down in front of the computer in an Excel spreadsheet and type all the information in. It is, to me, an absolutely heinous process. There's so much room for error. It's so difficult. So we want to replace, if ever they attempted to take out a slate, they need to talk to us about why, and we need to figure out a solution to that. And we're doing pretty well on that front. We're moving through the workflow. We aren't all the way there yet, but making really good progress on that. And then along the way, there's also a lot of data entry they do that we think that we can replace with basically AI analysis of images that they collect. So instead of writing down the entering into the app the species and genus of a coral, just take a photograph of the coral, and we'll automatically ID it. So that is basically the goal, is getting all of the data entry done in the app. And then, of course, that gives us the advantage that the data is standardized across the branches, immediately available. We can do cross-branch analysis really easily. It's just a so much better way of doing it. Then the other piece of technology I'm going to be talking about is an underwater camera system. So it's powered from that solar platform that I showed you at the beginning. It also sends data back up there, which then gets radioed to land and up to the cloud. So it's a connected platform which has its pluses and minuses. It's been really useful for a lot of development things. And I'll show you some of the work we've done with it. The platform contains a camera. It's actually multiple cameras. There's a stereo camera system in it. It has a hydrophone, so we have a lot of bioacoustic data, and also a temperature logger so that we keep track of the water temperature with it. Among its capabilities also is a 24/7 live stream, not exactly a scientific tool, but we thought, or it's a fantastic outreach tool, but it turned out that it was also picked up by a group of scuba divers who then started creating a spreadsheet of all the fish they saw in it, so then it becomes a really interesting biodiversity monitor, and you can look at the sort of the species accumulation and how many different species they saw. I think we're up to like 117, 118 different fish species observed on the camera. So the focus here, using AI to facilitate Coral Garden's work. And yeah, three main things is speeding up the workflow in the water when the garden is in the water, simplifying the data collection so we take steps out of what the work that they're doing when they're in the water, and also all sort of the new methods that we can potentially do with using the data coming off of the camera platform. My focus in this work, and something I really want to emphasize throughout this talk, is that I feel like any success that we've had has been because we are using AI methods, but we are always cognizant of the system that we're working in. And we use a lot of knowledge of that system, potentially local knowledge, but and also expert knowledge. There's a huge scientific community in Maria. And it's really combining the AI with that local knowledge to inform how we use the models, I think has given us the success that we've had. And the first thing is just remembering that what we're doing, which is, at the beginning I said we're doing coral reef restoration, but what we're really doing is we aren't restoring coral, we're restoring an ecosystem. And just keeping a whole ecosystem perspective on our work, I think is really important. So some of the things I'm going to talk about are ideas for using computer vision for extracting data about the state of the coral fragments, using photogrammetry and how we can make the photogrammetry workflow actually work for us. I'll talk about some of the problems we had and also the solutions we're developing. And then bioacoustic analysis. We have a lot of sound data from both an experiment, a restored site, and a nearby control site. And we have similarly experiment and control data, image data. And so how to extract data out of the images that is ecologically meaningful and can be used to help assess the health of the coral reef. This first part is just quick. A lot of this, I'm not going to go into a lot of the technical details, but there'll be a few places down the road where I do. The first part is just how are we going to use computer vision in the coral gardeners workflow. The coral gardeners are already taking a ton of in-water photographs. Some of it because coral gardeners as a whole has such an emphasis on imagery, so everybody's just like, why not take a photograph? But also some of it because, for example, the donor colonies the original coral fragments are taken from. In French Polynesia, the government requires that they be monitored, and they want photographs of the colony before it was harvested, immediately afterwards, and then every 6 months to make sure that it recovers okay. So, and when those photographs are taken, they have to be taken, the gardeners right now are entering species on the app. And so we have a huge, from historical data set of probably, I think, 2,000 or 3,000 donor colonies, all labeled. And so I think we have an opportunity there to build a tool which means that they won't have to do that in-water data entry. There's some issues around that. Species is an interesting concept with corals. It's pretty fuzzy. There's a lot of contention. Genus is well resolved. Species is not very well resolved, but I'm sure we'll work that out. And then in the nurseries, every few months, they swim along the ropes and monitor 20% of the fragments that are in the nursery. They actually 10% of the fragments, they measure 20% of the fragments. They assess for bleaching and overall health. All of that data is entered by hand in the iPhone app. Much better than doing it on the slate, but if you can just imagine swimming by and taking pictures, or potentially taking video as you swim by, and then automating all of that work, I think it can be made, again, much faster and much easier. And so I think there's a lot of progress to be made in that area. Photogrammetry. So photogrammetry, maybe it's probably not a word everybody here knows, but basically it means take a lot of 2D images around something. If I was going to do photogrammetry right here, I'd take a camera and take pictures around and over. And then, it's a pretty well-known thing these days and well established from a software point of view. You can basically take that pile of pictures and you want to make sure they overlap. And then you can just stitch them together, find common points between different images, and build a 3D model of whatever it is you photographed, which is a fantastic way of potentially of doing the monitoring of the coral reef. What we want to know about the reef, one of the main things for assessing reef health is what fraction of the surface area has living coral on it, dead coral, bare substrate, and a really important issue is macroalgae, seaweed. When coral reefs are damaged, for example, either by humans doing something or even just by a storm, some corals die or get stripped off in a storm. Usually, historically, what has happened is that the coral grows back. If you're in a place where there is a lot of nutrient load, then it's possible that seaweed will grow faster than usual and will take over that space. Or if you're in a place where there's been a lot of fishing and a lot of the big herbivorous fish or the big plant-eating fish have been fished out, then there's no fish to nibble the seaweed as it comes in. And then the reef will get taken over by seaweed. So in a lot of the world, reefs seem to exist in this sort of bistable state where they can either be covered in coral or covered in seaweed. And if they get kind of stuck in the coral state and it's hard to turn them into seaweed, but if through all of these impacts on the reef, you flip it to seaweed, it's really hard to get it back to coral. And so being able to monitor what people call benthic, which means ocean floor cover, but sand, substrate, coral, seaweed, et cetera, is a really, really important part of assessing. It's like the first order. How is your reef doing? If you jump in the ocean and you look at a reef and there's no living coral on it, you probably know it's not a very healthy coral reef. And if it's all covered in living coral, there's a good chance it's pretty healthy, although you need to have a healthy fish community. Otherwise, it's kind of like having a forest with no animals and birds in it. So anyway, classification off of the photogrammetry. We thought, we'll give the coral gardeners a photogrammetry model and a user interface, and they can go in and they can tag what different things are. That was a complete failure. Nobody, after they've been in the water all day, wants to sit down in front of a computer, and especially not use an awkward three-dimensional user interface, which if you're not used to manipulating, I have to say I found it very hard to do. So what are we going to do instead? Well, this is where AI can enter the picture, and there's this lovely project, fairly recently published, this project called Coralscapes. And it bills itself as the first general-purpose semantic segmentation dataset for coral reefs. Then you combine that with, they combined it, I didn't, they combined it with a tool called SegFormer, which is a transformer-based semantic image segmentation package. So semantic image segmentation basically means every pixel gets classified into a class. And the classes are just the different types of substrate that I was talking about, and actually quite a bit more detailed than that. Really important point of this is, I think the thing that really drove the innovation here is the data set. And that data set has thousands and thousands of images which have been by hand marked up as to what the substrate, what they are, is an incredible resource. And you look at the list of people who worked on it, it's massive. And for a lot of this work, I feel like getting high quality label data so you can get high quality results out of the model is a stumbling point. But what this does is it gives us the ability to take an image, a 2D image, and classify. Here's an example. Here's an orthomosaic on the left. So it's a bunch of images downward looking on the reef, stitched together into one big image. And on the right is the output from the classification model, the different colors of different classes. And the sandy color is sand, and the gray is rock, and the pink is coral, and the green is algae. And it's hard for you to tell, but when you look at this in close, it does a pretty good job. It probably does as good a job as if I was going through it, fairly quickly, because I've got a giant pile of images that I need to label. And it can go into more detail than that. It can actually label some of the corals, some of the branching corals down to the genus level. So we can get quite a bit of information about benthic cover from this. So then the question is, how do we combine the 3D model with the 2D image classification? And that's what you see has happened here. So the way you do that is you have this big pile of photographs that you've taken to build the 3D model. And on every single one of those images, you run that classification algorithm, so you have a benthic classification of every single pixel that goes into the 3D model. You can kind of intervene in the workflow, in the steps of building the 3D model, and you can tag every point that goes. First, basically you build a point cloud that is located in 3D space, and then you build a mesh off of that point cloud. You can tag every point in the point cloud with which source image it came, where in the source image it came from, and every point is generally from multiple source images. So that means you get multiple benthic classifications on every point in this model, and then you can vote and figure out what the most likely benthic class is, and then you can project that down onto the mesh or onto the vertex points in here, and then you can just run algorithms across that 3D model in order to compute things like the average level of coral cover or algae cover or things like that. Super useful approach. We've got a prototype working right now. Peter is working on getting, well, actually, everybody who's doing software is working on this. Mark is working on getting the photography part of it into the iOS app, because these were just done on photographs that somebody took in the water, not using the Reef app. And Peter is working on turning this into a pipeline that can run automatically, and Sophia is working on extracting the metrics from the 3D model. But we're really optimistic that we're going to be able to go from photographs to data about the state of the coral reef, basically fully automatically within the next couple of months. So I'm going to move on now to what we've done with AI to understand what's going on with the acoustic and image data that's coming off of the reef cam. So bioacoustics, there's been a bunch of work that other people have done, and I don't like reinventing the wheel. We're going to hear about perch, I believe, tomorrow, and there's perch and there's surf perch, and they've proved to be really useful to us. Been working with Conservation Metrics in Santa Cruz, who do a lot of bioacoustics work. They've been working on supervised methods. I've been working on unsupervised methods. But anyway, there's a lot of interesting things to do with bioacoustics. When I got into the bioacoustics, the first thing I did was just like, well, what does the reef sound like? I just think it's really important to actually know what know what you're analyzing, and then you'll have better ideas about how to go about doing smart things with your data. So I'm going to play a little bit of data for you. So scrapes and clicks and knocks, and they're all very short in time. That was daytime. Nighttime. There's some similar sounds in there, but they're pretty much buried in that sort of high-frequency popcorn, which I'm told are shrimp. And it's just super, super noisy at night from that. So I think I just moved slides by mistake. Yeah, anyway, I think I basically said all this. Yeah, it's, day and night are very different. There's fish sounds and there's invertebrate sounds. And a lot of the sound events are super transient. And then I'm going to talk mostly about unsupervised methods, but talk a little about the work that's gone on with supervised methods. So the day and night difference, which is also the fish and invertebrate difference, when you look at the spectrograms and read the literature about it, basically most of the energy of the fish sounds is below 2 kilohertz. Actually, most of it's below 1 kilohertz. And the snap, crackle, pop, that's all way up above that. So that's sort of like the first thing to realize. And then there's the fact that these fish sounds, they're short. And so that's kind of what we're working on dealing with, is these short bursts of sound. I mentioned perch, so bioacoustics model trained, what I read said it was trained on the songs of about 10,000 bird species. That might be wildly incorrect. It's getting a thumbs up. Good. And then some other clever people took a bunch of sort of reef sounds and used transfer learning to adapt the perch model to classification of coral reef sounds. And then from my conversations with people at Conservation Metrics, they were like, well, the classes on surf perch aren't so great, but the underlying model is really great. So you can take the underlying model and use it for embeddings, and then you can either produce your own set of labeled classified sounds, which is what Conservation Metrics did, but I don't have the time or energy to do that, so I went in an unsupervised way. So the basic workflow is take the sounds, low-pass filter it, and then segment it into discrete little bits. And those little bits are quarter, half second long typically. And the embedding algorithm says that I think it said it wanted 5-second sound chunks. So I just zero padded it and embedded it, and it seemed to work just fine. So then I computed embeddings, and then you do what you usually do when you have high dimensional embeddings and you want to do unsupervised methods, which is dimensionality reduction and clustering. And it produced about, around 20 different sound clusters. You can, there's a few parameters where you can make there be more or less, but around 20 seem to be typically what happened. So I'm going to play a few of the sounds, I think. Before I do that, you'll hear a sound, and then there's a short gap, and then there's another version of the sound. So these are like a few examples of the sound that was identified in the clustered sound chunks. So repeated, scrapey, weird sounds. I know you don't have a great description for them. Here's another one. So there's just single knocks. And then the last one that I have for you. Also single sounds, but a lot more low frequency content. That sounds like a fish beating on its swim bladder. So it seems like there's two main ways that fish make noises. That they either grind things together, like their back teeth, or they have like these mobile ribs which they can use to whack on the swim bladder, the bladder that they have in their bodies. And sometimes they combine them so you get this combination of, the grinding is called stridulation and drumming. And yeah, a lot of it I think is territorial. A lot of it I don't think we even know yet what species is making what sound. But then, if I'm interested in like the overall sort of diversity of fish life on the reef and the variety of different things happening on the reef in the fish community, I don't necessarily need to know who's making what sound. What I need to know is how many different sounds are there, and are the sounds on a healthy reef different than the sounds on a less healthy reef? And so that's kind of what I said change slide. Thank you. So this is just a quick view of the kinds of results. I picked out five of the classes and looked at how often, sounds per minute, these sound events happen. On the blue is the experiment site and the orange is the control site. And this should have actually been two plots. There's two things going on here. One is that if you actually stacked up all those bars, you would see that there's a lot more. There's about 1.5 times as many of these like loud sound events happening on the experiment site, which is where the restoration took place, compared to the control site. And the other is that the relative frequency of the different sound classes is different. So the fish communities in those two places are quite different, which is a really nice thing to know. Something else I really like about these unsupervised methods is that if I go to Fiji or if I go to Thailand, I can set up an experiment and control site and do this kind of comparison, and I don't need to know anything about the fish communities in those places. So, I mean, ignorance is bliss, perhaps, but it's really nice to have a methodology which is potentially widely deployable without having to have really specific knowledge about the fish community everywhere you go. Which I think just covered most of what I had on this slide. Yeah, there's clear difference between the restored and unrestored areas. The unsupervised methods transfer. Labels are really difficult, and even without the labels, I think there's a lot of really valuable information in the acoustic data. So I'm going to move on to the camera data. I'm just going to start with an image of the reef with I will say this is quite a lot more fish than usual. This is actually the first frame of a video that I'm going to show you in a minute. But this is the camera, where the camera was restored in the main restoration site on the north shore of Moorea. So most of the corals that you see in this image are ones that were planted by the coral gardeners. There's an image a little later on of the control site. This image also has this day and time just had very nice light conditions, so it produced this very beautiful slide. But the basic question I'm interested in is there a viable alternative to diver swarm transects for assessing the fish population? It's a lot of work to train someone up to know the fish well enough to be able to do an accurate survey. Having a diver in the water skews the survey anyway, because every time you get in the water, you see lots of fish run and hide. So you're not getting a great count anyway. And it's a lot of work to go out in a boat with the diving gear. It's just expensive to do that kind of monitoring work. So what I'm most interested in with monitoring is not like in the population level of an individual species, but is the overall state of the fish community. Because if you have an ecosystem that's got a healthy bottom layer, which is like the primary producers, the corals, and you've got a healthy fish community in it, you've probably got a healthy invertebrate community too, because the fish have to be eating something, and it's probably put those two things together. It's a good first cut at measuring overall ecosystem health. So things we're interested in is how many fish there are, how big are the fish, and what is the distribution of body sizes, because especially when there's been a lot of fishing, you tend to fish out the big things, and the big things do things that the little things can't. And do you have the right types of fish? I was talking about that earlier with the reef switching from coral to algae. If you don't have the herbivores in there, the reef is much more vulnerable to these sorts of state shifts. So the first thing, when I first looked at, heard about this and saw some pictures, I was like, oh, we can just sort of label these up with what species the fish are and build a model. But then like I said to you, we've seen like about 100 fish. Most of the fish you see are rare. And so to get training data for 100 different fish species when most of them are rare is going to be next to impossible. And then ecologically, I mean, somebody who's a fish ecologist might kill me for saying this, for understanding sort of overall ecosystem function, I don't think we need to know the identity of every single species out there. I'm much more interested in the functional groups. Do you have the herbivores? Do you have the different types of herbivores? Because there are the ones that eat the big leaf, the sort of the macroalgae, then there are the ones that nibble on the turf algae, and the ones that scrape off of the living and dead coral. There are all these different types of herbivores. And do you have a decent population of the omnivores and planktivores swimming around? Do you have corallivores? If you have corallivores, you probably have coral around too. So these just sort of basic functional groups I think are what we need to know. And we can understand those functional groups, especially in a place like Moorea, by digging into their 20 years of long-term monitoring data. And we can even correlate functional group with benthic cover, because they have co-located surveys done once a year for the last 20 years for that. So you can understand the correlation between for example, the level of herbivorous fish and the level of coral and algae cover in the system, and really identify which functional groups are important to the system. So that was kind of what I was thinking about when I was designing the computer vision model. Just the key really is just like, what are we need a big pile of labeled data, and what are the classes going to be? I want the classes to be things which are ecologically important, reasonably common so that you come out with a relatively balanced data set from your labeling, and also visually distinct, because if you define two classes, but the two fish are like next to impossible to tell from apart, that's not going to be much good. And there are some fish, like there's two different species of parrotfish that are common in Moorea, and even the fish experts can't really reliably tell them apart. So you definitely want to be smart about the classes that you define. So we labeled about 2,000 images. It was pain to do that. It was really hard to get people to do it. It is hard to do. You have to know the fish, and the images, your eyes hurt after a little while because the images are not easy to deal with. We fine-tuned the YOLO V8 object detection model, so just an off-the-shelf computer vision model. Fine-tuned it, we get about 85 to 90% precision and recall on the model, so it's doing pretty good. And then we just set it to work classifying big piles of images, and so we got a large fish abundance data set. Labeling. This is an image off the control site. Most of what you see here is seaweed. There's a few little patches of coral in here. You see there's boxes around the fish in here, but they're hard to see. You pull up an image, they're fairly big images. You pretty much have to zoom in and then scroll around, and then you see something, and then you think you found them all, and then you look again, and then you see another one. I mean, it's really time-consuming and difficult, and hopefully it is, in the end, worth the results. So I just wanted to show you this video because it's stunningly beautiful. And then it plays a second time with the fish detection model running on it. So it just gives you an idea of the kinds of data that this produces. If you look in the middle coming towards you. And we have no idea why, but after the eel passes, there's just a huge abundance of fish, way more than there usually are, for a couple of minutes. And if I stopped this on any frame and looked at it, we could go through, and you'd see that you'd see all the things that are right, you'd see all the things that are wrong, like the logo that was added by Coral Garda's Media House after the fact. The algorithm thinks it's a fish. Luckily, that's not there in the usual images. But this butterfly fish, it gets the butterfly fish really well because they're visually quite distinct. this it just says is a fish. I happen to know that's a parrotfish, and there's some others in here which it did get. This fish has got this little white spine down here, so when it's side on, it always correctly IDs it as a brown tang. But yeah, overall it does a pretty darn good job, and so then we can take the data from both the experiment site and control site and aggregate it together and look for trends. It does look like overall there are just a lot more fish at the experiment site, which is not too surprising. Over the course of about a year, there seems to be a decline in fish at the control site. We really don't have any understanding of why that's happening. Over here, this is the sort of the other fish, which are like the planktivores and the reef-associated fish, seem to be increasing at the experiment site. The herbivores are decreasing at the experiment site. The corallivores aren't really changing in level, even though a bunch of restoration work was done. But there are a lot more of them than there were at the control site, where there's very little coral, so that's not too surprising. One thing that I know about the corallivores, they're mainly butterfly fish, and butterfly fish are highly territorial. And it's entirely possible that by the time the camera was out there collecting this data, there was enough coral out there that basically the butterfly fish had reestablished territories. And apparently once they established territories, they defend them pretty well and they're pretty stable. So that could be why there wasn't a change in the level of corallivores. But that could also just be that could be a just so story that I'm telling you because it makes sense after the fact. So some of what we learned from doing this work, well, first thing is just labeling. Labeling is hard, it's time consuming. It requires you to really know quite a bit about the fish community or just to have a good eye. And it's hard to get people to spend very much time doing it. I wish that we could get out of having to, like with the acoustic data where we seem to have methods that are not going to require labeling. I wish we could do this without labeling. It might be that we can just isolate fish and then create like visual similarity buckets and do a similar thing with these images that we did with the acoustic data, and so not have to do all of that labeling work, which especially when you start, okay, we've got a model that works here in Maria, now we want to go to Fiji and do it there. We want to go to Thailand and do it there. It's different fish, different places, need more labeled data. And so that makes me question this. Something you would have seen in the video is that you can see there's a very highly camouflaged, I think it's a grouper on the left-hand side that the model does not pick up at all. Camouflage is a huge issue. We're really good with camouflage if the fish are moving. You pick up the movement. These models all work off of single images, and with no movement, you can't find them. So it'd be nice to develop some more sophisticated algorithms so that we can detect the camouflaged fish. I question single camera monitoring. There's a lot of heterogeneity and territoriality. So down here in this image, there's a small reef-dwelling fish called a humbug, black and white striped fish. It's taken up residence there, so probably at least half of the images have 1, 2, or 3 of them in it. If you turned the camera a little bit, you wouldn't see any of them. And so I think that probably figuring out how to deal with this issue that the reef is highly heterogeneous. You move 10 meters, it can actually look quite different. And yeah, we need to figure out how to deal with that. And also, this camera is connected back to the platform, and that's just a place, an opportunity for things to break. Working in the tropics is hard, working in remote locations is hard, and so having a tethered system like this is fine for some sort of flagship demonstration projects, but it's not a readily deployable system, so I'd love to figure out a way around that, and we're actually working on a much simpler short-time deployment battery-powered system, and so I imagine taking a restoration site like this and having like 4 or 5 of these cameras, putting them in, so you're sampling in multiple locations for maybe like 2 weeks, you probably have 2 or 3 weeks before the glass got too fouled, and you couldn't get good data anymore. And so I think that doing that, you could get a much higher quality data set. But even that, I think, would be a lot more reliable data and a lot easier to do than the kind of diver surveys that people are doing now. So I see a lot of problems, and that's partly because I'm a scientist and maybe because I'm English, and so I notice problems in things. but I also see a lot of potential for this, and I'm really excited to push this forward. So I think this is my last slide. I hope I've taken about the right amount of time. I've been 2.5 years, half time at Coral Gardeners. We've done a lot, we've come a long way, and we just have so many ideas of new things and different things that we can do. I think one of the hardest things is just staying on mission and making sure the work we do is relevant, making sure that it's focused on questions that are really going to affect the restoration work and make the whole project work better. The key to that, I think, is having deep knowledge of the system that you're working on, as well as hopefully, maybe not deep knowledge myself, but access to people who have deep knowledge of the technologies which are emerging, and combining them into tools which then also are designed to be scalable, deployable, and easy for people to work with. And then really underneath all of that is just the collaboration. Working with the people in the field, working with scientists throughout the community, working with technologists, just figuring out what's out there, what we could potentially use. And yeah, that's about the end of it. I also like to definitely thank our funders. OceanKind, our main funder, and I know Jason is here, and great to see him here. But it's been fantastic working with them, and it's been just an amazing project to get to work on, really inspiring. So thank you. And so, yes, I know it was kind of different than everything else, but I'm happy to answer questions.
Thank you so much. Any questions, Hugo?
I really absolutely love the breadth of the work. And I was curious if you've done anything with the third corner of the trifecta with the eDNA.
That is part of we have a project between Coral Gardeners, Conservation Metrics, and UC Santa Barbara, working out of the Gump Station, also funded by OceanKind, that is actively looking at eDNA. Yes. Don't have any results to share yet. We have a meeting in Santa Barbara at the beginning of September, but we're getting there. It's another technology. We've got to figure out how to use it and how to make it scalable, deployable. I'm really interested in a lot of DNA technologies, not just eDNA, but also like microbiome, population genetics for looking at the coral genomes and better understanding what's going on with them. I think DNA is going to be a huge part of our future. Yeah.
I guess I just question you. So it seems like there's so much stuff you can do, and in the 2.5 years, you've touched on a little bit about the bioacoustics. Where do you think, with like the current state of machine learning, where do you think, if you were going to have one more year, where would you point your lens? Is it? And then I was also thinking about the cameras that you're using, because I know lighting underwater is always an issue. Have you thought about using more hyperspectral cameras rather than just plain RGB type cameras?
We definitely looked into hyperspectral, but it's just expensive.
Yeah.
And yeah, the video I showed is lovely. And I was talking about potentially dealing with camouflage by looking at movement. But an awful lot of the time, the sun is shining, there's waves on the surface, there's these super intense caustics. And so dealing with the caustics in order to differentiate fish movement from light movement is a whole nother can of worms. But I know there are a ton of really smart people out there working on all this kind of stuff. I would love to see improved computer vision algorithms. I think that there's a lot of improvement that we can make there. And but yeah, if I was going to really start a new initiative there, it would be DNA focused. I think we need to understand the biology of these systems and what's going on in them as they are subject to climate change. I think that's going to be really, really important. The key to the restoration, if we're going to be replanting corals back out into the wild, it's what do we plant? That's, I think, the most important question to answer.
And do you draw upon people working in terrestrial environments? I know there's there's been a longer history of terrestrial restoration. How does that kind of impact your work?
I'd say in a lot of ways it hasn't that much yet. Yeah, we're much more connected into just because there is such a big, actually, and a very long history of people working. There's a good 20 years in the Caribbean of people working on restoration there, and so we've mainly been focused on people who've been working on other reefs around the world. Yeah, but definitely a place to look.
Other questions? Excellent. Thank you so much.
Yeah. Right,